Good evening to Nanda. Myself, Dr. T. Prabha. I am from Nanda College of Pharmacy, Erodu, Tamil Nadu. Today I am here to give a presentation on histone modification role in breast carcinogenesis and associated inhibition in breast cancer management. So normally, a cancer cell invasives and metastasis contribute to epigenetic modification. There are many epigenetics takes place in breast cancers, for example, DNA methylation and histone acetylation. So particularly, histone modification is one of the dynamic marker that is linked to the breast cancers. Histone changes can also support chromatin modulation, transcription, DNA repair, and genome stability. Since the epigenetic changes are reversible, epigenetic enzymes have gained a lot of interest as a potential target for anti-cancer therapy. Thus, there is a need to examine epigenetic alteration in breast cancer to define new markers and therapeutic targets. Why I chosen the study? The rationale behind the study is there is limitations in SIR study. For example, diverse thiazole compounds have reported as an anti-cancer drug. However, the SIR of 2-aminothiazole, which is linked with the MID and immune modifications, are not reported so far. And the next rationale is to find out the molecular target. So most of the studies are focusing their pathways rather than gene alteration in the breast cancer development, and also to improve the multi-target potential by applying the hybridization techniques with thiazole molecule by adding a number of pharmacophores. We can go for getting a potential target for treatment of breast cancer. So these things are under explored. So because of that, I have chosen this work. The aim of the study is the present study focus to design, synthesize, characterize, and pharmacological assessment of novel substituted 4-phenyl 2-aminothiazole scaffold, which contains amide and imine moiety that target histone epigenetic modification, particularly the genes like H3K9, H3K27, H3K4 in breast cancer through drug design and discovery via applying computational strategy. So this is how my workflow follows. First, I have synthesized the compound of 4-phenyl 2-aminothiazole compounds. So I have synthesized around 12 compounds by using different aromatic aldehydes and solvents. And after synthesizing the compound, I've gone for spectral characterization to confirm the synthesized compound by NMR and mass spectra. And then I have initially pharmacological assessments were undertaken by taking in vitro anti-inflammatory study and in vivo anti-inflammatory study. For example, in case of in vitro, then IBB denaturation and protease inhibition assays, and in case of in vivo, for a paragenine-induced paradigm method. So after completion of preliminary anti-inflammatory study, I have done the docking study by taking the protein data bank code of 1-ERE. This 1-ERE is the estrogen receptor, particularly expressed during the breast cancer type. Okay, and then after docking, the hit molecule, the lead compound will be taken for cell viability assay study by MTT assay on MCF7 breast cancer cell line. And coming to the results part, the in vitro anti-inflammatory study of the compound, for example, T3A, T3D, E, and so these compounds showed a 20% of inhibitions, while 82%, 76% when compared to the standard drug at 400 microgram per ml concentration. Further, the in vivo anti-inflammatory activity on bioedema inhibition, the compound showed 53% when compared to the standard drug diclofenac, whereas it showed 76%. So, when comparatively among all those compounds, this T3A compound showed noteworthy inhibition, which is near closer to the diclofenac compound. And then the same way, the docking score also somewhat good for T3A compound. So, based on the in vitro study, based on the in vivo anti-inflammatory study, and based on the docking score, we come up with go for TTSA by taking only this compound T3A compound. And on MTTSA on cell viability study, the compound T3A showed maximum cell viability at the concentration of range of 3.95 to 125 microgram per ml or in cell line. So why this cell line were chosen? Because of the gene size in H3K7, H3K24, H3K29. So these epigenetic histone genes are mostly expressed in MCF7 breast cancer cell lines. Coming to the summary and conclusion, currently there are a lot of anti-cancer treatments are undergoing for treating breast cancer. However, there is no particular medication to target the epigenetic modification. So MCF cells are often linked with epigenetic markers because they are best studied human breast cancer cell line and that exhibit significant alteration in cyst mastillation which directly impact gene expression and contribute to the development and the progression of breast cancer. According to our finding, a number of derivatives have notable anti-inflammatory and anti-proliferative activity when compared to the standard drug. So this insight gained from our study contribute to the ongoing efforts in discovery of and optimization of therapeutic agents for treating breast cancer via epigenetic modification. I am much privileged to thank all the patents and especially Dr. Muhammad Hanifa, Deputy President, Kerala State Branch, and all the organizing committee members for organizing such wonderful pharmaceutical research conference 2025. And thank you, one and all. Thank you for your listening.